इससे नेक्स्ट स्लाइड सबसे नीचे वाले से कुछ नहीं ना, ठीक है, इसके ऊपर प्रेजेंट तो करो।
Hello, I told you we are going online, so please.
Good morning to all respected professors and my dear friends. Myself, Som Thange. Our group seminar topic is autoimmune disorders. These are my group members: Nayan, Nikita, our class representative Harsh, Shivam, Puneet, Nancy, Nisarg, Sarth, and Rohan. So, what are autoimmune disorders? Autoimmune disorders occur when our body's immune system attacks our own cells, considering it as foreign invaders. Our immune system breakdown. So, in a healthy system, the immune system defends against pathogens. In autoimmune disorders, the system malfunctions, leading to inflammation, tissue damage, and dysfunction of affected organs. Prevalence affect millions worldwide with varying severity and impact on individuals' health. क्या आपको पता है क्या बहुत फेमस बॉलीवुड अभिनेता है जिनको माइस्थीनिया ग्रेविस है? हाँ, करेक्ट. What causes autoimmune disorders? So, genetics, environmental triggers, hormonal factors, and immune system dysregulation. In genetics, family history can be a risk factor, and certain genes can predispose some individuals for autoimmune disorders. In case of environmental factors, there are stress, diet, and other factors. And in hormonal factors, women are more susceptible for the autoimmune disorders. Maybe it's because of high level of estrogen. Classification of autoimmune disorders. There are mainly two types of autoimmune disorders: organ specific and system specific. In systemic autoimmune disorders, autoantigen present all over the body and autoimmune reaction mediated by autoantibodies. In organ specific autoimmune diseases, autoantigen are organ specific and autoimmune reaction mediated by T cells and autoantibodies. Uh, in case of organ specific diseases, in multiple sclerosis, brain is affected. Uh, uh, Hashimoto thyroiditis, thyrotoxicosis, primary myxedema. In all three diseases, thyroid gland is affected. In case of pernicious anemia, stomach is affected. In case of non-organ specific diseases, uh, in case of SLE, skin, kidney, and joints are affected. Symptoms of autoimmune disorders. There are some general symptoms: fatigue, joint pain, swelling, skin rashes, hair loss, fever, sensitivity to sunlight. Uh, Coming to how are autoimmune disorders diagnosed. So there are several steps in which a clinician will diagnose the autoimmune disorders. When patient comes to clinician, he will take first detailed medical history. After that, physical examination. After the physical examination, there are some initial blood test has to be performed. After that, auto antibody test. If there are some specific systemic involvement, then we will go for a targeted organ function test. Example: thyroid, liver, kidney, etc. If there are no such systemic involvement, then imaging studies will be done. example X-rays, ultrasound, MRI. After that, patient will be referred to the specialists for definitive diagnosis. If needed, biopsy can be done in case of diagnosis. Treatment of autoimmune disorders. In treatment, there are medications. In medications, patients are provided with immunosuppressants. Example steroids, DMARDs, biological therapies, TNF inhibitors. Lifestyle changes. Patient have to adjust with his or her diet. He should take anti-inflammatory diet. Exercise and stress management can be very helpful in the treatment of autoimmune disorders. Monitoring regular lab tests and specialist visits in case of rheumatologists. Conclusion: autoimmune diseases are complex and varied, but understanding their types and the organs or system they affect is crucial for early diagnosis and effective treatment. Both organ specific and systemic disorders have unique challenges but can be managed with proper care and medical support. Ongoing research is needed to better understand autoimmune diseases and develop more effective therapies. I would like to thank Ashwin sir and thank you everyone for listening to me.
Uh, Good morning, respected professor and my friends. My name is Nancy, and I am here to explain diagnosis of rheumatoid arthritis. First test is rheumatoid factor. This blood test checks for rheumatoid factor and antibody that many people with rheumatoid arthritis can eventually have in their blood, and uh, not all people with rheumatoid arthritis this test positive, and some people with this test positive can also not have rheumatoid arthritis, and some people test positive but have another disease. So this is not confirming test for rheumatoid arthritis. So which have further test which is anticyclic citrullinated peptide antibody test, anti-CCP test. This blood test checks for anti-CCP antibodies which appears in many people with rheumatoid arthritis. This test appears before rheumatoid arthritis symptoms, so we can early diagnose rheumatoid arthritis, and confirming this test also useful in confirming rheumatoid arthritis with RF factor, rheumatoid factor. However, it is important to know that some people have rheumatoid arthritis even with normal blood test. So now मैं एक स्टोरी बताना चाहती हूँ. मेरे कुछ करीबी फ्रेंड्स को रूमेटॉइड आर्थराइटिस है, तो उसको मतलब इन्फ्लामेशन की वजह से इन्फ्लामेशन के ज़्यादा सिम्टम्स दिख नहीं रहे थे, बट उसको रूमेटॉइड आर्थराइटिस था. तो इधर मैंने जैसे कहा कि रूमेटॉइड आर्थराइटिस का टेस्ट ये टेस्ट नॉर्मल भी आ सकता है, बट फिर भी रूमेटॉइड आर्थराइटिस हो सकता है. तो मतलब अगर टेस्ट में नहीं आए फिर भी रूमेटॉइड आर्थराइटिस हो सकता है, तो अर्ली डायग्नोसिस हो सकता है. Another test is complete blood count. This blood test measure different blood cells counts and can help diagnosis anemia. Rheumatoid arthritis, which people have rheumatoid arthritis, is common in anemia. Another test is erythrocyte sedimentation rate. This test measures inflammation in the body and monitors disease activity and response to treatment. C-reactive protein. This is another common test for inflammation that can help diagnosis rheumatoid arthritis and monitor disease activity and response to treatment. Now we are here to complications. Osteoporosis. In osteoporosis, bones get weak and fragile, and rheumatoid nodules. In rheumatoid nodules, some areas of skin which have inflammation in exterior part of the body, and dry eyes, mouth, infections, carpal tunnel syndrome. Here we are मतलब जिसको रूमेटॉइड आर्थराइटिस है उसमें कार्पल टनल सिंड्रोम होने की ज़्यादा हो सकता है, क्योंकि इन रूमेटॉइड आर्थराइटिस व्रिस्ट में इन्फ्लामेशन होता है. तो मीडियन नर्व को कंप्रेस होने की वजह से कार्पल टनल सिंड्रोम हो सकता है. तो जिसको भी रूमेटॉइड आर्थराइटिस है उसमें कार्पल टनल सिंड्रोम दिखने की मतलब ज़्यादा हो सकता है. हार्ट प्रॉब्लम्स, लंग डिसीज, लिम्फोमा. ट्रीटमेंट: anti-inflammatory medicines for pain relief and lower inflammations. Corticosteroids, which is disease modifying anti-rheumatic drugs that helps to slow or change progression of disease. Biological response modifiers and genus kinase. These two are also DMARDs, and if your disease does not respond to initial therapies, these medications target specific immune message and interrupt the signal, helping to decrease or stop inflammation. In genus kinase, which are also DMARDs, send message to specific cells to stop inflammation from inside the cell. These medications may also consider if your disease does not respond to initial therapist. Here are our source, and thank you so much to our professor and friends.
Good morning, everyone. My name is Sarth Patil, and I will be sharing information with you all on systemic lupus erythematosus. Not so very common autoimmune disorder, but it is necessary for all of you to know about it. The topics I will be covering is what is actually SLE, prevalence, causes, pathogenesis of SLE from the biochemical aspect, symptoms of SLE, and finally challenges in living with systemic lupus erythematosus. So, what is systemic lupus erythematosus? It is a chronic autoimmune disorder where the immune system attacks the body's own tissues and organs. It is systemic. In the name, the systemic suggests that it affects multiple parts of the body such as skin, kidneys, heart, and joints. The most commonly affected organs include the skin, the kidneys, heart, lungs, blood vessels, and the brain. Prevalence. In global prevalence, there are 20 to 150 cases per 100,000 people worldwide. Gender disparity, women are more affected to SLE. There is a ratio of 9:1, very high ratio, but generally in autoimmune disorders, women are mostly more prevalent. This is due to the hormonal differences between the two genders. Age group. It is most common in women of childbearing age between 15 to 45 years. Ethnic variations, there is a higher prevalence in African, Hispanic, and Asian populations. Geographical differences include more common in urban areas and regions with advanced diagnostic tools. Disease severity, the ethnic groups with higher prevalence often experience more severe symptoms and complications. The pathogenesis of SLE, this flowchart shows the process for pathogenesis of SLE. The genetic predisposition, female gender, environmental factors like silica exposure, smoking, or taking oral contraceptives may lead to basically two factors: defective apoptic cell clearance and nucleic acid exposure, and increased formation of NETs. I'll come to this later. Many of you might know from our physiology syllabus what are apoptic cells. Anybody willing to contribute? Yes, programmed cell phenomena. Actually, IFN signature is the interferon stimulates the overexpression of certain genes called the IFN signature genes. These IFN signature genes then lead to the extra formation of the T cells and the B cells and provide an immune response against the tissues of the body. Thus, activation of the T cells helps the B cells to produce more antibodies. And this is the B cell activation. Now, to the increased formation of NETs, NETs are actually neutrophil extracellular traps which are formed on the B plasma cells. So, when the neutrophils come to attack the B plasma cells, the NETs, the traps, trap the neutrophils, and therefore the B plasma cells are not phagocytosed, and therefore they long-lived plasma cells in the BM, and so there is an increased formation of the antibodies. Causes of systemic lupus erythematosus are basically categorized into genetic, immune regulation, and environment. The genetic predisposition may take certain individuals more vulnerable to SLE. Environmental triggers like sunlight, infections, or certain medications can also trigger SLE. As a sort of treatment or providing relief to the patients affected with SLE, they are advised not to expose their skin to direct sunlight. Hormonal factors, women, especially those in the reproductive years, are more likely to develop SLE. Immune system dysfunction, the body's immune system mistakenly targets its own cells and tissues. Symptoms, the common symptoms of SLE, like other autoimmune disorders, include fatigue, joint pain, and swelling. A very particular symptom for SLE is the butterfly rash, as you can see here. The first picture shows it in its initial stages, and the second one is when it gets severe. It is a very distinguishing feature of SLE. Fever, photosensitivity, which is sensitivity to sunlight, hair loss, and mouth ulcers. One more distinguishing feature is the Raynaud syndrome. Actually, it is very common syndrome, not just observed in SLE. Many of you might have felt very cold fingers in case of winters. In winters, your fingers get extremely cold. Some of you might have felt that. So it is basically because the capillaries in your fingers develop spasms and they contract. Therefore, there is a less heat release there, and so your fingers turn extremely cold. It is a very common syndrome, so most of you might have experienced it. The organ-specific symptoms for SLE include kidneys. The kidney damage specifically the kidney damage caused due to systemic lupus erythematosus can be referred as lupus nephritis. Heart, the chest pain or inflammation of the lining of the heart, and nervous system, seizures, headaches, or memory problems. I will discuss the challenges in living with systemic lupus erythematosus. Since there are no such treatments yet discovered for systemic lupus erythematosus, the most we can do is provide relief to the patients by various procedures. Unpredictability. Symptoms can flare up and subside, making daily life challenging. We previously saw the pathogenesis of SLE, so we kind of have an idea that how the increased production of T cells and B cells cannot be prevented, and therefore how the symptoms can sometimes flare up and cause increased distress to the patient. Chronic fatigue, a significant issue for many SLE patients. Organ damage, long-term lupus can cause serious organ damage, especially to the kidneys and the heart. Mental health, coping with chronic illness and can lead to anxiety and depression. These are my references, and I thank you all of you for patiently listening to me. I call my friend Shivam for explaining the diagnosis and treatment of the disease.
Hello. Am I audible? So good morning, respected professors and all my dear friends. My name is Shivam Agrawal, roll number 7 from group 3, and I will be discussing about the diagnosis and treatment of SLE. Learning objective, first of all, the diagnosis, in which we will see the lupus band test, anti-dsDNA test, and some other tests, and then we will see the treatment, in which pharmacological and non-pharmacological treatment will be there, and then conclude. First of all, the lupus band test. In this test, the clinically normal skin should be taken for the biopsy from the area or the area near the rashes, and then the biopsy is stained with the direct immunofluorescence. And if the test is positive, then immunoglobulins are complement to it, and they are found at dermoepidermal junctions. As sir previously mentioned, SLE is a large systematic disease, and occasionally it can be organ dominant. So we possess some diagnostic challenges for this. So this test is not very sensitive for SLE, but it is highly specific for the SLE. Now, anti-double stranded DNA test, in which we firstly take the blood serum from the patient with suspected SLE, and it is mixed with the anti-double stranded DNA, which acts as an antigen for the test. If the antibodies are present in serum of the patient, they will bind to this antigen DNA, and the measure of the level of the antibodies will be done by using the ELISA technique. Some other laboratory tests are there, like complete blood count, urine analysis, skin biopsy can be done, erythrocyte sedimentation rate, and the kidney biopsy, liver biopsy also we can use in organ specific very affected. Then now we will come to the treatment for the SLE. The current goal of the treatment is to achieve the disease remission or state of low activity. Like in first of all, we see the non-pharmacological treatment. In this, lifestyle modifications are required. As Sar previously mentioned, SLE is it is a highly photosensitive disease. So we should avoid the sun exposure to the patient, do regular exercise, and should take healthy diet, and regular monitor your blood pressure levels, kidney function and urine protein levels also. In pharmacological treatment, WHO generally guided to take the immunosuppressant drugs for the patients of SLE. The point here is to mention that we know that SLE is generally very common in women. So women should women with suspected SLE should not take the estrogen only containing oral contraceptive. They can use the progesterone only containing pills, or combined oral contraceptive, or intrauterine devices also can be used, but only estrogen containing should not be used. The conclusion is that the SLE is very complex and variable disease, so it requires a multidisciplinary approach to the treatment and diagnosis. Early recognition helps in preventing irreversible organ damage and improving the long-term outcomes. And the prognosis has improved with advancements in treatment, but ongoing research is essential for better therapies and understanding of the disease. Thank you so much. Then I call my colleague Nayan Pandey for continuing the next topic.
Good morning to all the professors and my dear friends. So myself Nayan Pandey, roll number 63, and I am going to present on the topic diabetes type 1, and I will be covering in these subtopics: symptoms, complications and treatment. And as we know that diabetes is caused because of the autoantibodies are developed against the beta cells in the pancreas which secrete insulin. So these are the symptoms, and the three cardinal symptoms of diabetes are the excessive thirst, which is called polydipsia, and excessive hunger, polyphagia, and polyuria, which is excessive urination or frequent urination. And since glucose is lost in the urine, so it also takes with it excess water, and because of that there is polyuria. And since the fluid is lost from the body, so the thirst centers in the hypothalamus are triggered, and which leads to polydipsia. And since there is lack of insulin or deficiency of insulin, so because of that the glucose is not utilized by the cells, so they feel like they are starving for energy. So the body also triggers the hunger centers, and thus excessive hunger is also seen. And the excess glucose in the body fluids that also absorbs the fluids from various tissues such as the lens of the eyes, and this leads to change in the shape of lens and certain characteristics, which leads to blurred vision. The excess glucose in the blood also it impairs the healing abilities of the body, and also impairs WBC function, which leads to slow healing and of cuts and sores. And there is also fatigue. There is a yeast called Candida albicans, which naturally lives in the human body and in certain tissues like vagina. So when there is excess glucose, so it provides favorable condition for this yeast to grow, and therefore there are vaginal yeast infection also. Now moving on to the next slide. These are a few complications. Excess glucose in the blood vessels lead to it binds with various proteins, and it leads to the damage to the vessels, microvascular damage or macrovascular damage, which can lead to cardiovascular diseases. And if these vessels, if in the retina there is an inhibitor of CAT1, which is carnitine acyltransferase 1, so which is responsible for the transport of acetyl-CoA into the mitochondria, and responsible for beta oxidation. Now if malonyl-CoA is not there, so it will not inhibit the CAT1 enzyme, and because of that beta oxidation will take place, and as byproduct ketones are synthesized, ketone bodies such as acetyl-CoA, acetyl-CoA, and these ketone bodies lead to diabetic ketoacidosis. And it is characterized by the fruity odor in the breath of the patient. So it is the ketone bodies are responsible for that. Now coming to the diagnosis of diabetes type 1, the HbA1c test is done, the glycated hemoglobin test, which detects the average blood glucose levels for the past 2 to 3 months, and a level greater than 6.5% or higher indicates diabetes. And random and fasting blood glucose tests can also be done. The fasting levels are normal is 80 to 100, and if it is greater than 110 or 120 to 140, so it indicates diabetes. Since it is important for the clinician to distinguish between diabetes type 1 and type 2, so the doctor will also advise to do the blood test to check the presence of autoantibodies against the beta cells, which will indicate diabetes type 1. Now coming to the treatment, the insulin therapy is must for the treatment of diabetes type 1, and there are types of insulin like rapid acting insulin or long acting insulin. Rapid acting initiates its action within 15 minutes or so, and the long acting insulin takes several hours to initiate its effects. And there are many methods of giving the insulin therapies, such as injections or insulin pumps. And this here the image depicts an insulin pen. There are continuous glucose monitors which is inserted into the body, and this continuously monitors the level of glucose in real time. And blood sugar monitoring is done, and balanced meals and regular exercise will stabilize the blood sugar. Now coming to the current researches and advances in the treatment, artificial pancreas system is developed, and it involves combining the insulin pumps with continuous glucose monitors. So in this picture, as we can see, this is the continuous glucose monitor, and it monitors the level of blood glucose in real time. And when the glucose levels increases, through a computer controlled algorithm, it sends signals to the insulin pump. Then insulin pump releases certain quantities of insulin which is required into the blood, and then the effect in the patient is seen. Now, the current researches are also going on gene therapy to restore the beta cell function, on stem cell research, and pancreas and islet cell transplants. Recently, in year 2023, a study was done on a patient using the transplantation of CIPSCs derived islets. CIPSC stand for chemically induced pluripotential stem cells. Pluripotential stem cells, as we know that they have the capability to differentiate into multiple types of cells, and such as they can differentiate into the beta cells. So the cells are the adipose tissue cells are extracted from the patient, and then they are chemically reprogrammed. So initially in conventional, what they used to do was the induced pluripotential stem cells were made by genetic overexpression of the genes. But what they did was that they used certain molecular chemicals for reprogramming the cells, and thus they made chemically induced pluripotential stem cells. Then the other task was to obtain efficient insulin from these cells. So obtaining efficient beta-like cells. Then after that, they tested for the safety and efficacy of these chemically induced pluripotential stem cell islets in non-human primates, and then they observed that there is no long-term tumorigenicity, and it is safe for the patient. So afterwards, then they implanted these islets into the abdominal anterior rectus sheath. Previously, conventionally, they used to infuse these islets into the hepatic portal vein, which was inducing immediate blood-induced immune response against these cells. So that was leading to lesser survival of these. So what they did they implanted it into an extrahepatic site into the abdominal anterior rectus sheath, which proved to be increasing their survival and no long-term tumorigenicity, and it was also measured by the increased secretion levels of C peptide, which was used as an endogenous marker of the insulin secretion. Then then they seen the effect in the patient of these cells. So after 75 days, they observed that there is an insulin independence post transplantation. So that means this treatment was effective. And also the glycated hemoglobin levels decreased to about 5% after these implantations. And the time in target glycemic range also was greater than 98%. Time in target glycemic range means that the total time in a day in which the glycemic range or the sugar levels of the blood are within the normal range. So that also increased to 98%. So that was this was the experiment, or this was the research which they did, and that was proved successful. So these are my sources. Thank you.
Good morning, everyone. Myself Rohan Arya, and today I'm going to talk about vitiligo. Uh, vitiligo is a common depigmenting skin disorder. It is an autoimmune disorder which is characterized by selective loss of melanocytes, which typically results in non-scaly, chalky white macules. And there are various types of vitiligo, which are first one is generalized vitiligo. In this type of vitiligo, there are small macules all over the body, and the second is segmental, which affects only one side or one area of the body, and the third is mucosal vitiligo. In this type of vitiligo, mucosal membrane is affected, and genitals are also affected. And the fourth one is trichrome vitiligo. In this type of vitiligo, the affected area, the central part of the affected area appears smooth and white, has a smooth and white appearance, and the peripheral part has a lighter color of the natural skin tone. As you can see that the central part has a smooth white color, and the peripheral part has a lighter color of the natural skin tone. Now the fifth one is focal. It is a rare type of vitiligo in which the white patches are restricted to one place, and the white patches don't spread. And the sixth one is a universal type of vitiligo. It is also a rare type of vitiligo in which the skin loses pigmentation over 80% of the body. Now, how to diagnose vitiligo? To diagnose vitiligo, the doctor will ask about the patient's family history, and the doctor will also perform a physical exam. The physical exam includes close evaluation of the skin, and in individuals who have lighter skin, the doctor uses a Wood's lamp, which is also known as black light, which is an ultraviolet light. So when the doctor puts this lamp, the vitiligo patches appears bright. As you can see in this image, these vitiligo patches are appearing bright in under black light, and this is Wood's lamp. And the doctor may also recommend some tests, which are first is blood test to check for other autoimmune disorders, and eye exam to check for uveitis and inflammation of part of the eye that sometimes occurs with vitiligo. And the doctor may also take a skin biopsy to check for missing melanocytes from that depigmented area. Now comes melanogenesis. Now melanocytes produce melanosomes, which store melanin, and the number, size and distribution of melanosomes determines skin color. So this is the pathway of melanin synthesis. In this pathway, the key step is the activation of microphthalmia associated transcription factor, which leads to the downstream activation of these enzymes, which are tyrosinase, TRP1 and TRP2, which are essential for the synthesis of eumelanin and pheomelanin. So eumelanin is a brown or black pigment that gives color to the skin, hair and eyes, and pheomelanin is a red yellow pigment found in human skin, hair and cultured melanocytes. Now, pathophysiology of vitiligo. Vitiligo is commonly known as multifactorial polygenic disorder and has complex pathogenesis. It is commonly associated with both non-genetic and genetic factors. Various theories have been proposed about its pathogenesis, but the exact etiology is still unknown. So these are some theories which have been proposed. First one is neural mechanism. In this neural mechanism, a neurochemical mediator usually destroys the melanocytes. Now the second is oxidant and antioxidant mechanism. An intermediate or metabolic product of melanin synthesis causes the destruction of melanocyte. In intrinsic defect of melanocyte, there is intrinsic defect in the melanocyte which causes the destruction of melanocyte. The fourth is autoimmune or cytotoxic mechanism. There is an alteration in humoral or cellular immunity that causes the destruction of melanocytes. Now these are some genetic factors that lead to vitiligo. First one is multiple genes involved. Research has identified over 30 genes associated with an increased risk of vitiligo. So variations in these genes can increase the risk of vitiligo. and the second is major histocompatibility complex genes. The HLA genes within the MHC region are strongly linked to vitiligo development. So variations in these genes also will lead to vitiligo development. The third is polygenic nature. Vitiligo is considered a polygenic disease, meaning multiple genes interact to increase the risk of vitiligo. Uh, Now, fourth is family history. Individuals with a family history of vitiligo have a increased risk of developing vitiligo. Now, these are some environmental factors. The first is sun exposure. Excessive exposure to the ultraviolet radiation from sunlight can potentially trigger vitiligo. And the second is trauma or injury. Skin trauma, burns, or abrasions at specific sites can sometimes lead to the development of vitiligo patches. Third is stress. Psychological stress is considered a potential trigger for vitiligo. Fourth is chemical exposure. Contact with certain chemicals like hair dyes, cleaning products, or skin whitening agents may trigger vitiligo. Now, the role of oxidative stress in vitiligo. Oxidative stress is considered to play a crucial role in activation of autoimmune response related to vitiligo. Melanin synthesis inside the melanocytes is the major intracellular stressors that produce reactive oxygen species. So under normal physiological conditions, these reactive oxygen species are converted into non-toxic forms by some antioxidative factor like nuclear erythroid factor 2. But under pathological conditions, these reactive oxygen species don't get converted into non-toxic forms and causes the destruction of melanocytes. So, as you can see in this figure, so UV radiation and chemical oxidants are considered as exogenous stressors, which lead to the accumulation of reactive oxygen species in keratinocytes, which eventually lead to the apoptosis of melanocytes. And we can also see that by accumulation of reactive oxygen species, there is increased release of cytokines which activate cytotoxic cells, which also leads to the apoptosis of melanocytes. And the accumulation of reactive oxygen species can also be by DNA damage. So when there is accumulation of reactive oxygen species, it will eventually lead to apoptosis of melanocytes. And when there is increased concentration of hydrogen peroxide inside the mitochondria, that leads to alterations in DNA, which causes abnormal level of reactive oxygen species that leads to destruction of melanocytes. So how does this oxidative stress causes destruction of melanocytes? So oxidative stress causes melanocytes to produce antigens which activates autoimmune response, and the second is accumulation of oxidative stress causes release of chemokine which attracts immune cells to the skin lesions, leading to destruction of melanocytes. So oxidative stress can be treated by antioxidants like uric acid, glutathione, vitamin C and E, and it can also be prevented by antioxidant enzymes like catalase, reductase, thyroidoxin. So difference between vitiligo and albinism. So vitiligo is an autoimmune disease, while the albinism is a genetic disease. So vitiligo results from immune system killing melanocytes, while albinism is present from birth and occurs because patient is inherited a faulty gene required for making melanin. And in vitiligo, the patches appear smooth white, and in albinism, the patient's skin, hair, and eyes appear extremely pale. Although there is no treatment of vitiligo, but we can bring back some color and reduce the spreading of vitiligo. So these are some treatment options. First is phototherapy. In phototherapy, the UV light is incident on the affected area, which induces the melanocytes to produce more melanin. And the second is gene therapy. Scientists are working on gene therapy for vitiligo development. And the third is melanocyte transplantation or surgical method. In this technique, melanocytes are taken from donor skin biopsy and are transplanted into vitiligo patients. The fourth is topical calcineurin inhibitors. They work by inhibiting the activation of T cells which attack the melanocytes in vitiligo patients. The fourth or fifth is nanotechnology applications in vitiligo treatment. Nanotechnology applications are being used to increase the effectiveness of drugs in vitiligo treatment. Now, these are some herbal remedies which are effective in vitiligo treatment. First is ginkgo biloba, cucumis mellow, and third is chelin, and fourth is polypodium leucotomus. So these are some psychological and social impacts of vitiligo. The individual who has vitiligo has low self-esteem and it is socially isolated, depression and anxiety, relationship difficulties, career challenges. Now comes conclusion. Vitiligo is a chronic skin disorder that produces white patches on skin. The exact etiology of vitiligo is still unknown. It may start at any age and affect any part of the body. There is no fixed cure of vitiligo. It is not a harmful disease. It causes psychological problems like tension, embarrassment, and loss of self-confidence. Thank you.
ये इससे आगे बढ़ेगा, इससे और ये पॉइंट करके आगे किससे बढ़ता है? हाँ सही है, सही है, सही है.
Good morning everyone, my name is Harsh Dakar and today I am going to discuss about multiple sclerosis. First we will discuss about the normal immune response and autoimmune response. In normal immune response, when foreign antigen enter in our body, antibodies are formed against it and will destroy it. But in case of autoimmune response, antibodies are start forming against our self cells and will start destroying it. Let us discuss about the introduction. Multiple sclerosis is a demyelinating disease of central nervous system which includes brain and spinal cords. In multiple sclerosis, demyelination happens when the immune system inappropriately break, attack and destroy the myelin, which make communication between neuron break down and ultimately leading sensory, motor and congenital problems. Let us discuss about some of the facts which are related to multiple sclerosis. Multiple sclerosis was first described in 1868 by Jean-Martin Charcot. The name multiple sclerosis refers to numerous scars that develops on white matter of brain and spinal cord. Let us discuss about the definition. Multiple sclerosis is a demyelinating disease in which insulating cover of nerve cells in brain and spinal cords are damaged. Let us discuss about incidence which are related to multiple sclerosis. Approximately 2.5 million pupils are affected with multiple sclerosis worldwide, but most of the cases are seen in women, approximately 3 times more than in men. Now we are discuss about the etiological factors which are related to multiple sclerosis. It can be idiopathic, it can be genetic factor, it can be environmental, it can be due to infection or maybe due to vitamin D deficiency. Now we will be discuss about the pathogenesis of multiple sclerosis. As we know that the myelin sheath, which is a protective covering on axon which is found by oligodendrocytes in central nervous system. Like blood-brain barrier allow only some of the specific molecules to get enter, such as T cells or B cells. T cells, when these T cells cross the blood-brain barrier attached to a specific type of protein that are present in the myelin. Now after that, T cell activated by myelin and change to endothelial cells to express more receptors. After that, it allow immune cells to get easily bind to receptors. These specific T cells produce cytokines, interleukin 1s, interleukin 2s, which dilates the blood vessels. After that, we will discuss about the type of multiple sclerosis. So there are mainly 4 type of multiple sclerosis depend on sign and its symptoms. First, there will be relapsing remitting multiple sclerosis in which bouts of autoimmune attack happening after months or even in years and causing an increase the level of disabilities. Secondary progressive multiple sclerosis, which is similar to RRMS, but after some time immune attack become constant, which cause a steady progression of disability. After that, primary progressive multiple sclerosis, constant attack on myelin, which cause steady progression of disability over a person's lifetime. And last, progressive relapsing multiple sclerosis, constant attack, but both superimposed, during which disability increase even faster. Now we will be discuss about clinical manifestations in multiple sclerosis. Charcot's neurological trait of multiple sclerosis, which includes nystagmus, dysarthrias and intense tremor. Other clinical manifestations are dysphagia, loss of vision, blurring of visions, muscles weakness and spasm, ataxia and Lhermitte's signs. These are my sources. Thank you. And now next continue by the Nikita.
Good morning everyone, my name is Nikita Meena, roll number 65 from group number 3, and I'll continue with the multiple sclerosis under the subheadings causes, diagnosis and treatment. First of all, we will talk about the causes. The causes of the multiple sclerosis is the combination of genetic predisposition and environmental triggers that affects the autoimmunity and results in loss of myelin. So, if we talk about the genetic aspect, multiple sclerosis itself is not an inherited disorder, but having a parent or sibling with multiple sclerosis can increase the risk of getting multiple sclerosis, since hundreds of genes and gene variants combine to create vulnerability to multiple sclerosis. For example, genes encoding for HLA-DR2. As usual, it is more common in females, and both T cells and B cells are affected in this. So now let's move towards the environmental factors: smoking, obesity, stress, virus exposure and low sun exposure can increase the risk of multiple sclerosis. For example, the Epstein-Barr virus is consistently linked with the development of multiple sclerosis. And if we talk about the sun exposure, research indicates that people who spend more time in sun and having more level of vitamin D are less likely to get multiple sclerosis, because vitamin D may regulate the immune system. So if we look at this picture, we can infer that the region far away from the equator is more prone to multiple sclerosis relatively than the area at the level of the equator. So let's move to the diagnosis. As there is no specific test for the multiple sclerosis, but we can do some other tests to confirm or rule out the diagnosis. For example, lumbar puncture, which is also known as the spinal tap, in which we obtain a cerebrospinal fluid and examine it for proteins and inflammatory cells associated with the disease. Next, MRI of the optic nerves and OCT. You can look at this MRI scan of the multiple sclerosis, in which the red arrow indicates the brain lesion. And next one is evoke potential test, in which we place electrodes on the skin and send painless electric signals to measure how quickly and accurately the nervous system responds to the stimulation. Now let's move towards the treatment. So let me tell you, I will talk about the treatment very briefly, because there are different types of treatment for the different types of multiple sclerosis. So we will talk about the common ones. As there is no permanent cure for multiple sclerosis, but there are treatments that can reduce the number and severity of the relapse and delay the long-term progression of disease. For example, corticosteroid, because they are anti-inflammatory and immunosuppressive. Intravenous immunoglobulins and cyclophosphamide, plasma exchange, which is also known as the plasmapheresis. This treatment involves separating plasma cells from the blood cells and replacing it with the albumin solution. Some other treatments including managing symptoms, spasticity, fatigue, pain and bladder issues, physical therapy that can help with mobility, strength and coordination, cognitive rehabilitation therapy, and lifestyle modification, which includes vitamin D, diet, exercise and stoppage of smoking. And some of the emerging treatments include gene therapy and stem cell therapies. So these are my sources. Thank you.
इसको क्या करना है इसका नीत गुप्ता के नाम से नीचे नीचे टॉपिक का नाम है वो टॉपिक का नाम भूल गया अरे वो अपना सोराइसिस नीचे यहाँ पे जाइए थोड़ा नीचे हाँ नहीं हाँ यही है Good morning, respected Professor Sir and my dear friends. So, so here clinical case is given, and here a 28-year-old male patient presented to a dermatologist with red, raised, itchy and scaly patches of the skin present on the scalp, in the front of the knee and behind the elbow from the last 3 months. He used to drink the alcohol and the doctor advised him a cream and a drug which is applied directly and on the affected skin to slow down the skin cell growth, and also advised him for the UVV phototherapy. With the time patient condition improved. What do you think? From which disease this person is suffering? What are the cause for this? So what are your thinking? Anyone can guess what is the disease? Yeah, this is psoriasis. So here this is psoriasis by me and my roll number is 76. Psoriasis is an autoimmune disease and it is a condition in which skin cells build up and form scales and itchy dry patches typically seen on the elbow, knee and lower back associated with the white scaling on the scalp. ये जो है 3% पॉपुलेशन में देखने को मिलेगा, यानी कि कॉमन टाइप है जो कि दिखता है, और क्रोनिक डिजीज यानी कि लंबे टाइम तक ये जो पर्सन है उसमें दिखेगा और इसका कोई परमानेंट क्योर नहीं है, इसे रिड्यूस कर सकते हैं बट ये अगेन डेवलप हो जाएगा. दिस कंडीशन टेंड टू गो थ्रू द साइकिल्स फ्लेयरिंग फॉर अ फ्यू वीक और द मंथ देन subsiding for a while. Can occur at any age but more common at the age of the 15 to 13, as you see here. The 28-year-old male patient was given, and new cells are produced 8 times faster than normal, but the rate at which old cells slug off is unchanged. So here the new cells जो हैं वो बहुत जल्दी प्रोड्यूस होंगी, तो सेल मास जो है वो बहुत ज़्यादा ग्रो हो जाएगा और वहाँ पे ब्लड जो है वो भी अकुमुलेट हो जाएगा, तो यहाँ पे रेड कलर का जो है वो भी देखने को मिलेगा, और यहाँ पे जो वेल डिमार्केटेड लाइनिंग होगी जो कंडीशन बन रहा होगा उसमें. Here are the some site of the psoriasis lesions. You can see here scalp is one side, elbow, gluteal cleft, umbilicus, ears, nails, genitalia and knees. And there are some symptoms of the psoriasis and the types. Common signs and symptoms of the psoriasis include itching, burning or soreness, cyclic rashes that flares for a week or month and then subside, dry cracked skin that may bleed, presence of the rashes. I will show how the psoriasis develop in the further slide. And here the cause of the psoriasis, as I discussed earlier, psoriasis occur when the skin cells are replaced more quickly than the usual, and the reason is not well known, but a researcher think that it is due to the problem with the immune system, as it is as I told earlier that it is an autoimmune disease and new skin cells are produced in the deepest layer of the skin. और ये कॉन्टेजियस डिजीज नहीं है, यानी कि यहाँ पे छुआछूत वाला ऐसा कुछ मामला नहीं है.
here you can see no psoriasis in the first condition, in the psoriasis in the second condition, and on the top you can see here red color and skin folds scales you can see, and this is because यहाँ पे next slide में there are some etiological factors and due to that यहाँ पे केराटिनाइज जो होते हैं हमारे सेल जो है स्किन का, वहाँ पे जो केराटिनाइज स्क्वेमस एपिथेलियम होता है उसमें हाइपरप्लेसिया हो जाएगा, एंड ड्यू टू दैट यहाँ पे जो है वो साइटोकाइन का फॉर्मेशन ज़्यादा होने लगेगा, और यहाँ पे जो साइटोकाइन का फॉर्मेशन है वो फर्दर एक्टिवेट करेगा टी सेल्स को, टी सेल्स फर्दर प्रोड्यूस करेंगे साइटोकाइन, और यहाँ पे इम्यून सिस्टम जो है वो अच्छे से वर्क नहीं कर पाएगा, एंड यहाँ पे ऑटोइम्यून डिसऑर्डर होगा जिसमें एपिडर्मिस का इन्फिल्ट्रेशन एंड केराटिनोसाइट प्लोरीफरेशन होगा। यहाँ पे फर्दर जो ब्लड वेसल्स हैं वो डायलेट हो जाएंगी एंड वेस्कुलर एनगॉर्जमेंट हो जाएगा, और एपिडर्मल हाइपरप्लेजिया एंड इम्प्रॉपर सेल मैचुरेशन जो है वो सिल्वरी स्केल ऑफ द स्किन को लीड करेगा, एंड फर्दर यहाँ पे जो ब्लड है वो अपीयर होगा। डायग्नोस्टिक एवोल्यूशन ऑफ द सोराइसिस, जो डॉक्टर है वो सबसे पहले हिस्ट्री लेगा इफ देयर इज एनी जेनेटिक हिस्ट्री ऑफ द पेशेंट, बिकॉज द मेन रीजन्स आर द genetic regions or maybe unknown regions. Physical examination will be done, skin biopsy will be done to find if there is any other condition, if there is any other condition. So their diagnostic evaluation में skin biopsy भी की जाएगी and blood and radiography to rule out the psoriatic arthritis. And there are some etiological factors that is unknown or genetic, or there are some other factors which can जो कि इसकी स्पीड को तेज कर सकते हैं, लाइक स्ट्रेस, स्मोकिंग, एल्कोहल, ट्रॉमा, ओबेसिटी, हार्मोनल चेंज, क्लाइमेट, ऑटोइम्यून डिजीज, एंड देन देर आर सम ट्रीटमेंट ऑफ द सोराइसिस. Psoriasis treatment aim to stop the skin cell from the growing so quickly and to remove the scales. Options include creams and light therapy and phototherapy and oral or injected medications. Some topical therapies are given here and light therapies also. Here the light therapy, the aim of the light therapy is to reduce the growth of the cell, because UV light can decrease the rate of the cell growth here. And it is from my side. Here one more thing is that कि जो विटिलिगो है एंड जो सोराइसिस है उनमें डिफरेंस क्या होता है. लाइक if a person is having vitiligo then he can develop psoriasis, and if a person is having psoriasis then he can develop vitiligo. Why? Because the genetic condition that lead to vitiligo and psoriasis are very much common, and that is why if a person is having vitiligo he should उसे थोड़ा सा अपने आप में ध्यान देना चाहिए कि वो कहीं कुछ नया चेंज तो नहीं देख रहा है अपनी स्किन में. सोराइसिस वाला जो पेशेंट है उसको भी देखना चाहिए कहीं कुछ नया चेंज तो नहीं है, क्योंकि हो सकता है कि वो दूसरा डेवलप कर लें एंड देन उसको मोर दिक्कत हो. इसमें सोशल स्टिग्मा भी होगा, क्योंकि यहाँ पे जो पर्सन है उसके लिए स्किन में दिक्कत है. तो यही, थैंक्स.